Welcome back to the Deep Dive, ready to explore some fascinating science and tech news. Today we're looking at the latest from February 10th and 11th. Buckle up, because we've got a lot to unpack, starting with new developments in tuberculosis treatment, all the way to the intricate world of single-cell multi-omics.
Let's dive right in.
Tuberculosis. It's a bit shocking that in 2025, it still claims over 1.3 million lives each year.
Unfortunately, it remains a significant global health challenge. The good news is researchers are making headway in developing new vaccines and therapies.
So what makes tuberculosis so hard to treat?
Well, the bacteria, Mycobacterium tuberculosis, is incredibly resilient. Think of it as a master of disguise. It evades our immune system by hiding inside our very own cells.
So it's playing hide and seek with our immune system.
Exactly. That makes it very difficult to target with typical antibiotics.
And that explains why developing effective vaccines has been such an obstacle.
Yes, the current vaccine, BCG, has limited effectiveness, especially in adults. The complexity of tuberculosis and the bacteria's ability to adapt and evolve rapidly makes it hard for our immune systems to mount a strong, lasting defense.
That explains the significance of these new vaccine candidates.
Absolutely. Scientists are exploring several approaches, including vaccines that aim to trigger a more targeted immune response, and others trying to prevent those latent infections from becoming active. Plus, there are new drug combinations and immunotherapies that are showing promise on the treatment front. These therapies essentially boost the body's natural defenses against tuberculosis.
It's encouraging to see progress on multiple fronts. Could be a real game changer. Now let's shift gears to something called SIMO. It sounds futuristic.
It does, doesn't it? SIMO stands for Spatial Integration of Multi-Omics Single-Cell Data. Imagine having a comprehensive molecular blueprint of a single cell. This blueprint includes its genes, proteins, modifications, and even its location within a tissue.
Wow, it's like a detailed 3D map of a cell's entire molecular landscape. What makes this spatial context so important?
Well, cells don't operate in a vacuum. Their behavior is influenced by their surroundings, their interactions with other cells, and their location within a tissue. SIMO provides an unprecedented look into these complex relationships.
I can see how that could be revolutionary for disease research.
Exactly. Imagine diagnosing diseases like cancer with pinpoint accuracy by seeing how disease cells differ from healthy ones in their molecular makeup, interactions, and spatial organization. This could pave the way for personalized treatments tailored to each person's unique cellular profile.
It's like taking personalized medicine to a cellular level. Speaking of breakthroughs, researchers have found immune cells that actually fuel the growth of an aggressive brain tumor. That's fascinating and a bit counterintuitive.
It is. This finding, thanks to RNA sequencing, could be significant for creating targeted therapies.
Hold on. Isn't the immune system supposed to fight cancer? How can immune cells actually help a tumor grow?
Cancer is very sneaky. It's a master manipulator of its environment, including our immune systems. In this case, the tumor tricks specific immune cells, attracting and reprogramming them to support its growth and survival.
So instead of attacking the tumor, these immune cells are actually nourishing and protecting it. That's quite a survival strategy.
It certainly highlights the complexity of cancer. But now that we know these cells are playing a role, we can develop therapies to block their recruitment or even reverse their tumor-promoting activity.
So a whole new avenue for treatment. Now, how about we explore the world of artificial intelligence, specifically generative AI, which is making waves in psychology?
Generative AI is fascinating. It can create new content like text, images, and even music. Researchers are exploring its potential to completely change how we approach mental health care.
How could generative AI be used in psychology?
Imagine AI-powered tools that generate personalized therapy plans, customized to an individual's needs and preferences.
That would be revolutionary. But how would it work?
Well, an AI system could analyze a patient's symptoms, history, and personality to create a tailored treatment plan. This plan could suggest specific therapeutic exercises, coping mechanisms, or even personalized meditation scripts.
That takes personalized medicine to a whole new level. Are there any other possible uses for this technology in the mental health field?
Absolutely. Generative AI could also help develop early diagnosis tools. Think of an app that can analyze speech patterns or writing to detect signs of depression or anxiety before they even become obvious. Early intervention is key.
It all sounds incredible, but what about the ethics of using AI in mental health? It definitely raises some serious questions.
You're right. Addressing those ethical concerns head-on is critical. Ensuring responsible and ethical use of AI and prioritizing patient privacy and data security are paramount. Transparency is key, and patients should always understand how AI is being used in their care.
This is clearly a field that needs careful consideration and a commitment to ethical implementation. The potential benefits are huge, but responsible development is crucial.
The future of mental health care is likely to be heavily influenced by AI, and it's our responsibility to ensure that its benefits reach everyone equitably.
Switching gears to genomic medicine, there's exciting news from Israel.
LifeBit and Cephes have partnered up to create a secure platform for clinical genomic research in Israel. Their aim is to unlock breakthroughs in personalized medicine by studying Israel's very diverse population.
This sounds like it has the potential for some major advancements.
Imagine the possibilities of studying the genomes of such a diverse population. This platform will allow researchers to safely access and analyze huge amounts of genomic data, leading to deeper insights into diseases and personalized treatments. It's a win for both scientific progress and patient privacy.
That's a powerful combination.
Yeah.
This initiative really puts Israel at the forefront of personalized medicine research. I'm excited to see what discoveries emerge from this partnership.
This collaboration embodies the power of teamwork and data sharing in accelerating scientific progress and improving health outcomes for everyone. Now, let's change gears again and explore a variety of research making headlines today.
Starting with a study exploring the potential of etravirine, which is an HIV drug for treating liver cancer. This seems like a great example of drug repurposing.
It is. Researchers have discovered that etravirine disrupts the cell cycle of liver cancer cells, essentially inhibiting their growth.
It's remarkable how a drug developed for one purpose might hold the key to treating a completely different disease. This really highlights the importance of exploring all options in the fight against cancer.
Sometimes the solution is right there waiting to be discovered.
Next up, a study using AI to predict COVID-19 cases. You'd think the pandemic is behind us, yet research and analysis continue.
This study utilizes a long, short-term memory model. It's a type of AI that excels at analyzing data over time. By feeding the model factors like vaccination rates, mobility data, and past infection rates, researchers can predict future COVID-19 cases with impressive accuracy.
This AI model could be a game changer for public health planning.
Absolutely. Imagine authorities being able to anticipate surges, allocate resources more effectively, and implement timely interventions to minimize the spread of the virus. AI has the potential to be a powerful tool for safeguarding public health.
It's all about leveraging data to make informed decisions and protect communities. Next, we have a study that's using key TCT scans to predict how lung cancer patients will respond to immunotherapy.
Immunotherapy has transformed cancer treatment, but not everyone benefits equally. This study utilizes radiomics, which extracts information from medical images to analyze those PTCT scans.
So by analyzing these scans, researchers can predict who's likely to benefit from immunotherapy.
Exactly. They've identified specific features in the scans that correlate with immunotherapy response, helping doctors make more informed treatment decisions and provide truly personalized care.
Tailoring treatment to each individual, a major step toward precision medicine in cancer care. AI seems to be popping up everywhere in medical research. Here's another study that uses it to identify targetable changes in lung cancer images.
AI is a rock star when it comes to pattern recognition, often surpassing human capabilities. This study trained an AI algorithm to analyze histological images of lung cancer tissue. The goal: to pinpoint specific genetic alterations in cancer cells that could be targeted with already existing therapies.
So the AI is essentially learning to see cancer in the same way a pathologist would, but with more speed and accuracy.
That's a great way to put it. This is personalized medicine in action. By pinpointing the specific genetic weaknesses of a patient's cancer, doctors can choose the most effective treatment. It showcases AI's power in driving the shift toward more targeted and personalized cancer care.
From AI, let's move on to a study that's mapping T-cell dynamics to molecular profiles. I think it's safe to say that T-cells are vital players in the immune system.
T-cells are like the special forces of our immune system. They identify and destroy infected and cancerous cells. This study uses cutting-edge techniques to track T-cell movement and behavior, linking it to their molecular profiles.
It's like creating a detailed roadmap of T-cell operations within the body.
Exactly. This knowledge is essential for understanding how the immune system works and how we can harness its power to fight disease. Imagine being able to develop new immunotherapies that enhance T-cell activity against cancer or other diseases.
This type of research is truly pushing the boundaries of our understanding of the immune system and paving the way for new treatments.
The immune system is remarkably complex, and every discovery gets us closer to understanding and manipulating its intricate workings. Welcome back to the deep dive. We're picking up with a study that's looking at how heterologous fusions impact chromosome organization within mammalian cells.
Heterologous fusions.
Yeah.
What are those exactly, and why should we care about their effect on chromosomes?
Well, heterologous fusions happen when segments from different chromosomes get joined together. This can happen naturally or be induced in the lab. These fusions can really shake things up, disrupting the normal organization and function of chromosomes, and potentially leading to cellular abnormalities.
So this study is examining how these chromosomal mix-ups actually play out inside the nucleus.
Exactly. Using advanced imaging techniques, researchers were able to visualize the 3D structure of chromosomes. This allowed them to see firsthand how heterologous fusions change their arrangement within the nucleus.
So the spatial arrangement of chromosomes is important.
Absolutely. Think of the nucleus like a bustling city and chromosomes as distinct neighborhoods. Each neighborhood has its specific function. Heterologous fusions disrupt this organized system, creating chaos within the cell. Understanding this cellular chaos provides insights into things like development, disease, and even evolution.
So this research is giving us a peek behind the curtain of the nucleus, revealing just how complex these cellular processes are.
Every new study adds another piece to the puzzle. Shifting gears to Africa, there's a study highlighting the work of the African Animal Breeding Network.
Their aim is to improve livestock productivity and sustainability in Africa, a critical endeavor considering the challenges of food security and climate change.
I couldn't agree more. The network uses various approaches, including genomics and selective breeding, to improve livestock resilience, productivity, and disease resistance. They're focused on breeding animals that are better suited to African conditions. Traits like heat tolerance, disease resistance, and higher milk or meat production are key.
This initiative really showcases how science can be used to tackle real-world problems, improving both livelihoods and food security. Combining scientific knowledge with local expertise is crucial for creating sustainable solutions.
It really is. Let's circle back to immunology for a moment. There's a study mapping T-cell dynamics to molecular profiles. This research uses something called behavior-guided transcriptomics to connect T-cell behavior with their underlying molecular makeup.
Behavior-guided transcriptomics, that sounds pretty complex.
It is a revolutionary approach. It lets researchers observe the behavior of individual cells in real time while simultaneously analyzing their gene expression patterns. It's like having a window into the inner workings of T cells. You can see how they move, interact, and how those actions are reflected in their gene expression.
This research is revealing the intricate dance between cells and molecules within the immune system.
And understanding that dance is critical for figuring out how to create effective immune responses against various diseases. Let's move on to a study on CRISPR-Cas12a. This gene editing tool has really transformed genetic research.
CRISPR allows for precise gene editing, but there's always that risk of off-target effects where the tool cuts DNA in the wrong places. This study is looking at a factor that might influence CRISPR-Cas12a's accuracy, the flexibility of the DNA near the target site.
Picture DNA as a flexible chain. A rigid chain is less likely to bend and interact with the CRISPR machinery in the wrong spot, but a more flexible chain can wiggle around and bump into the CRISPR complex accidentally, even if it's not the intended target.
So DNA flexibility can impact how accurate CRISPR is.
Exactly. By taking DNA flexibility into account, scientists can design more accurate and efficient CRISPR systems. This can help minimize those unintended genetic modifications, and that's essential for ensuring the safety and effectiveness of CRISPR technology, especially as it moves closer to clinical applications.
This research is helping to fine-tune CRISPR, paving the way for its full potential in treating genetic diseases and developing new therapies. What's next?
Next, we'll delve into a study that explores breakage-fusion bridge cycles in cancer cells. These cycles contribute to genetic instability, which can make cancers more aggressive and harder to treat.
Breakage-fusion bridge cycles. How do those work?
Imagine a chromosome breaking in two. Normally, the cell repairs the break, but sometimes those broken ends fuse to the wrong partner, creating an abnormal chromosome. When this abnormal chromosome replicates, it can lead to even more breakage and fusion events. It's like a cascade of genetic chaos.
Doesn't sound good for the cell.
Not at all. These cycles can amplify cancer-promoting genes, fueling tumor growth. This makes treatment more challenging. But by understanding these processes, we can develop strategies to disrupt them, potentially slowing down tumor evolution and leading to better treatment outcomes.
So, this research is providing a glimpse into the inner workings of cancer and highlighting the challenges we face in treating this complex disease.
Now let's explore temporal lobe epilepsy, a neurological disorder that causes seizures. This particular study focuses on the molecular mechanisms behind the condition, especially a process called ferroptosis.
Ferroptosis, that's a new one. What is that exactly?
Ferroptosis is a type of cell death. Think of it as cell death driven by iron-dependent lipid peroxidation. Researchers are still trying to figure out its role in various diseases. This study is investigating whether ferroptosis is involved in temporal lobe epilepsy. They're looking for specific biomarkers and potential therapeutic targets related to ferroptosis in patients.
So this research could open the door to new treatment approaches for temporal lobe epilepsy.
Exactly. By targeting the pathways involved in ferroptosis, we might be able to develop therapies to prevent or reduce seizures. This highlights how basic research can lead to real-world clinical applications.
From epilepsy, let's move on to autism spectrum disorder, or ASD, a neurodevelopmental condition that affects communication and social interaction. This study is focusing on a specific type of ASD that's caused by mutations in the transcription factor 4 gene, or TF4.
This research is trying to understand how mutations in TF4 contribute to the development of ASD. They've used gene network analysis to identify pathways that are disrupted in individuals with TF4 mutations. Think of it like a complex network of interacting genes. When TF4 is mutated, it throws a wrench into the flow of information within that network, leading to downstream effects.
So this study is mapping out how those TF4 mutations disrupt gene networks, ultimately leading to ASD symptoms.
And by understanding those molecular pathways, researchers can start to identify potential targets for therapeutic intervention. This offers hope for more effective treatments for ASD down the road.
Unraveling the genetic and molecular underpinnings of complex conditions like ASD is essential for developing targeted therapies. What else is new in the research world?
Well, there's a study that's focusing on identifying the genes that drive specific traits. They've introduced a new method for prioritizing what are called effector genes. Those are the genes directly responsible for those observable traits.
They're pinpointing the genes that make us who we are, from our physical characteristics to our susceptibility to certain diseases.
It's like finding a needle in a haystack. Our genome has thousands of genes, but only a subset are directly involved in specific traits. This method combines different types of information, including genetic data, gene expression data, and functional annotations. This allows them to paint a clearer picture of which genes are driving those particular traits.
This integrative approach seems to improve both the accuracy and confidence of their predictions, a significant step forward in understanding the genetic basis of complex traits.
Definitely. This knowledge has implications for a wide range of fields, including personalized medicine and even agricultural breeding. Now, let's explore the world of mitochondria, the powerhouses of our cells. This study looks at hexokinase I, an enzyme involved in glucose metabolism, and its interaction with mitochondrial VDAC1 and VDAC2.
VDAC1 and VDAC2, what are those, and why is their interaction with hexokinase I important?
VDAC1 and VDAC2 are proteins found in the outer membrane of mitochondria. They act as gatekeepers, controlling what goes in and out of these cellular energy factories. Hexokinase I, on the other hand, is involved in the initial steps of breaking down glucose. This study discovered that hexokinase I directly binds to VDAC1 and VDAC2, suggesting a close connection between glucose metabolism and mitochondrial function.
So all of these proteins are involved in how our cells produce energy.
Exactly. This interaction might be crucial for regulating energy production, cell signaling, and even cell survival. Understanding these molecular interactions could lead to insights into metabolic diseases and potential new drug targets.
This research is really peeling back the layers of complexity within our cells. It's fascinating.
It really is. Now let's discuss a study that uses machine learning to predict breast cancer recurrence. They're using algorithms to analyze clinical data and predict the likelihood of both local and distant recurrence.
So they're developing a tool to determine if a patient's breast cancer is likely to come back, and if so, where it might spread.
Exactly. That information is vital for guiding treatment decisions and providing truly personalized care. The machine learning model is trained on a massive amount of patient data, including tumor characteristics, treatment history, and genetic factors. It learns to identify patterns that are associated with different recurrence outcomes.
So it's like a sophisticated pattern recognition tool that can help doctors make more informed predictions about a patient's risk of recurrence.
It's all about using the power of machine learning to analyze complex clinical data, paving the way for more personalized and predictive medicine.
AI is changing the face of healthcare, giving doctors and patients more information to make better decisions for improved outcomes. Now for something a bit different. A study that's using deep learning to automatically detect emesis.
Emesis, or vomiting, is a common side effect of medications and a symptom of various diseases. This study developed a deep learning system that can accurately detect vomiting in Suncus marinus, their shrews.
Shrews. Why shrews? And why is automatic vomiting detection so important?
Shrews are often used in research because they're small, easy to handle, and have short lifespans. Being able to automatically detect and measure vomiting helps researchers evaluate the safety and effectiveness of new drugs or study how diseases progress.
So it's a tool that can streamline research and improve the accuracy of data collection.
Exactly. Technology is playing an increasingly important role in enhancing scientific research. Speaking of technology, let's look at a study focused on developing a new imaging technique for spinal surgery. They've introduced a standalone ultrasound-based system that can create detailed 3D images of the spine.
Sounds like a significant advance in spinal surgery. What can you tell us about this new technology?
Think of it as a GPS for spinal surgery. This ultrasound system gives surgeons a clear 3D view of the surgical field, allowing them to navigate the complex anatomy of the spine with greater precision and safety.
This has the potential to really change how spinal surgery is performed, leading to more accuracy, fewer complications, and better outcomes for patients.
Technological innovations like this are driving progress in surgical procedures, benefiting both the surgeons and the patients. Now, let's talk about morphological profiling. It's a technique used to study the shape and structure of cells. This study is demonstrating how self-supervision can enhance morphological profiling by unlocking more powerful image representations.
We talked about self-supervision earlier. Can you remind us how it works and how it applies to image representations?
Self-supervision is a type of machine learning where the algorithm learns from the data itself without needing explicit labels. Image representations are essentially mathematical ways of encoding the visual information in an image. This study uses a self-supervised learning algorithm to extract meaningful features from those cell images, allowing for more accurate and detailed morphological profiling.
So it's using AI to dig deeper into the structure and function of cells.
Precisely. Now let's discuss a study that focuses on the Bacillus cereus group. These are bacteria known to cause food poisoning. This research is investigating the antiviral defense mechanisms of these bacteria.
Why is it important to understand how these bacteria protect themselves from viruses?
Understanding how bacteria fight off viruses can give us clues about new ways to fight bacterial infections. With antibiotic resistance on the rise, we need to explore alternative approaches. By studying how bacteria defend themselves against viruses, we might be able to identify targets for new antibacterial therapies.
This research is crucial in the fight against bacterial infections, especially as antibiotic resistance becomes a more pressing issue.
Staying one step ahead in the evolutionary arms race against bacteria is key to developing effective treatments. Now, let's talk about a study that sounds like something out of science fiction, a digital twin brain simulator.
A digital twin brain simulator. That sounds incredibly futuristic. What is that exactly?
It's a simulator that uses electrocorticogram data from primates. This data allows them to monitor consciousness and even simulate virtual interventions. It's like creating a virtual brain that mimics the activity of a real brain.
What are the potential applications for something like this?
The possibilities are vast. It could be used to study consciousness, brain disorders, develop brain-computer interfaces, or even test new drugs and therapies in a virtual environment before moving to human trials.
That's a major breakthrough in neuroscience and AI, opening up a whole new world of possibilities for understanding the brain and developing new treatments.
Welcome back to the final part of our deep dive into the frontiers of scientific discovery.
It's incredible how much groundbreaking research is happening every single day.
It really is a testament to our human drive to understand the world around us.
Absolutely. So let's jump right back in. We've got a study that revisits morphological profiling. Remember we discussed this earlier.
Yes, This research is exploring how self-supervision can take morphological profiling to the next level. Morphological profiling is a technique scientists use to analyze the shape and structure of cells.
Right. And self-supervision is all about algorithms learning from the data itself without needing us to explicitly label everything.
Precisely. In this study, they're applying self-supervision to images of cells. So instead of needing humans to label each image, the algorithm figures out those key features on its own.
It's like the algorithm is creating its own learning system based on the patterns it observes.
Exactly. This allows the algorithm to create powerful representations of those images, basically encoding all that visual information. It's like giving the algorithm a high-powered microscope to explore the world of cell morphology.
Incredible. I could see how this could revolutionize the field of morphological profiling.
Absolutely. It allows us to analyze huge amounts of data much faster and could even reveal subtle patterns that we might miss otherwise. Now, shifting gears a bit, there's a study focusing on the Bacillus cereus group. Those are bacteria that are known to cause food poisoning.
Oh, yes, I remember. So this research is looking at how these bacteria defend themselves against viruses. Why is this so important, especially for food safety?
Well, understanding the defense mechanisms of bacteria is critical for developing new strategies to combat them. And with antibiotic resistance becoming a bigger problem, we need to explore every possible avenue. By learning how bacteria fight off viruses, we might be able to find new targets for therapies.
It's like studying the enemy's playbook to figure out their weaknesses.
Exactly. This research could pave the way for new antibacterial therapies that specifically target those viral defense mechanisms. Now, for something that seems straight out of the science fiction movie, a digital twin brain simulator.
A digital twin brain simulator. That sounds incredibly advanced. What is it exactly?
Researchers have actually developed a simulator that uses electrocorticogram data from primates. This data is used to monitor consciousness and even simulate virtual interventions. Think of it as creating a virtual brain that acts like a real one.
It's mind-boggling. What are the potential applications for this type of technology?
The possibilities are pretty much endless. We could use it to study consciousness, understand brain disorders, develop brain-computer interfaces, and even test new drugs or therapies in a virtual environment before moving to human trials.
That's a game changer for neuroscience research. Well, that wraps up our whirlwind tour of some of the latest breakthroughs in science and technology. From advances in tuberculosis treatment to the mind-bending world of digital twin brain simulators, it's incredible to see the progress being made across so many different fields.
It's truly inspiring to see the power of human curiosity and innovation at work.
As always, we encourage you to keep exploring and learning. The world of science is constantly evolving, and there's always something new to discover.
Absolutely. Stay curious and join us next time for another deep dive into the fascinating world of science and technology.